Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries in programs like this. We'd like to thank the Anna Gordon Getty Foundation and Camilla and George Smith, whose generous support made this episode of Lunch Break Science possible. Here with us today is Leakey Foundation Baldwin Fellowship Scholar and grantee Habiba Chirchir. Habiba, thank you so much for being here with us on our 30th episode and just past Lunch Break Science's 1-year anniversary. Sorry, go ahead. Habiba is joining us from West Virginia, where she is an associate professor of biology at Marshall University and a research associate in the Human Origins program at the Smithsonian National Museum of Natural History. Her research has taken her all over the world to look at fossil collections. She's been to South Africa to scan fossils using a high-resolution micro-CT scanner. And the first image was of the Max Planck Institute in Germany. And then to her home country in Kenya to look at species of early Homo and Australopithecus. Before we hear from Habiba, if you are watching us live on YouTube, Facebook, Leakey Foundation Live, or Twitter, you can post questions for Habiba in the chat, and she will be answering them live during the episode. The earlier you get those questions in, the more likely she will be able to answer them. So Habiba, my first question for you is, your research mainly focuses on trabecular bone. What is trabecular bone?
Well, trabecular bone is a type of bone that you find mostly in the ends of long bones where we have joints, and you find it also in some of the flat bones and vertebrae. So what you're looking at in that picture is in the femoral head, and you see a nice where you see a nice lattice of white bone on the inside. That's where we usually find bone marrow. And the trabecular bone is also referred to as spongy bone. So if you hear someone talking about spongy bone, that's trabecular bone.
So what do changes in the robustness of bones tell us about the behavior of our ancestors?
Yes, so there are two things that I would like to say first before I get into what that can tell us about human ancestors. One is that we talk about robusticity of bone and we talk about gracility of bone. So when we talk about bone being robust, we refer to it being thicker, being denser. When we talk about gracility of bone, we are referring to its lightness, its low bone density, if you will. Now, when we look at our fossil ancestors, our prehistory, we see that from early on, as far as 3 million years with the Australopithecines, you see a robust bone or high bone density. And then over time, especially in the last 10,000 years, you see quite a change in which we have less bone density, not just in trabecular bone, but also in what's called cortical bone, which is the bone that forms the mid shaft of our forms the shafts of our long bones. The other thing that we can think about in reference to robusticity of bone is that when we compare, for example, our close living relatives, the chimpanzees and ourselves, living humans, we see that we can measure how strong the bone is at the mid shaft of, say, our thigh bones. So if we look at a chimpanzee thigh bone and we cut it in section, you see that the amount of cortical bone that's in the thigh bone, as well as that you find in the humerus, which is the forelimb, they're relatively even. And that's because chimpanzees are quadrupeds, so they're using the forelimbs and the hind limbs almost, those limbs are experiencing the same amount of loading. So you see an even distribution of bone. So not very robust, it's fairly robust, but evenly robust. But when we look at humans, because we are walking bipedally, right, the two limbs, we see that our femoral, our thigh bones tend to be quite robust. Okay. Then the other thing that I would like to take note of, based on that previous picture that you just showed, is the amount of trabecular bone that we find in our hand bones. So if you take a section through your, say, your middle finger at the joint, and you look at the spongy bone or trabecular bone that's in there, across different species, across chimpanzees, across our human ancestors and ourselves, we see that a chimpanzee will have very robust trabecular bone, while a modern human will have less, or will have gracile trabecular bone. And the other fossil ancestors varied a little bit, and we'll talk about that when we get to the research component of my work.
Well, I look forward to getting to that. But I mean, obviously, you didn't start your career kind of thinking like you're gonna specialize in trabecular bone. How did you or when did you become interested in pursuing science as a career?
Well, I started, well, as you mentioned, I am from Kenya, and I did my undergraduate degree at the University of Nairobi in anthropology. And part of that, my education was doing an internship at the museum in Nairobi, at the National Museums of Kenya. And I was very lucky to have met some graduate students, American graduate students, that I worked with and worked under. And in that process, I came up with a senior thesis that was related to some of their interests, some of my interests on looking at the outside of bone, taking measurements of bone. How do they compare between different species? How do you compare? Do they compare between different human populations? And thereafter, I went on and applied for graduate school and came to the US, and that's when I continued that kind of studies of comparative studies of long bones through my master's. And it's naturally, if you're looking at how, or my interest was looking at how activity patterns affect bone at the gross level, at the external level, and the natural progression was to look at how the inside of bone will change. So we know, okay, well, the outside of bone, the cortical bone will change if you strain it. It will grow denser, but what about the inside? And so that's how I ended up in studying trabecular bone for my PhD, and I continue to study the same even now.
So, two of your early, hold on a sec. Two of your early, fellowships that supported you were the Leakey Foundation's Baldwin Fellowship and Wenner-Gren's Wadsworth Fellowship. How did that impact your career?
Right. I was very, very lucky and privileged to have got those awards. First, the Wadsworth Fellowship from the Wenner-Gren to start to do my master's, or start the master's and then go on into the PhD. And then after that, for the PhD, I had the Baldwin Fellowship from the Leakey Foundation. And those were really instrumental because without most graduate school, of course, will offer a scholarship package of some type, but these fellowships really boosted my ability to, have a decent stipend and be able to concentrate with my studies and not worry about, well, do I need to hold a graduate teaching assistantship as well as going on with my studies. So those were really impactful in that way, and I'm ever so grateful to the Leakey Foundation and the Wenner-Gren.
Well, we've shared a link in the chat to learn more about the Leakey Foundation's Baldwin Fellowship, as well as the Wenner-Gren's Wadsworth Fellowship. So be sure to check those out and see if you may qualify for them. My next question is, what were some of the first collections that you had the opportunity to work with?
Little bit in time to my undergraduate studies that I was talking about. That's what I wrote my senior thesis on. And the first collection was a modern human collection, actually, that's housed at the museum in Nairobi. And this was, a group of skeletons of people that were doing farming and people that were pastoralists. And that was my first experience studying bones. Thereafter, as I went on to graduate school, I became exposed or studied more modern human collections. And of course, at the culmination of my graduate studies was a study of fossils. So some of the fossils that I've studied are of Australopithecines from South Africa, Australopithecines from Kenya, And more recently, actually, if I can move on to a recent work that's been funded by the Leakey Foundation, is the study of early Homo from Kenya. And that's an ongoing project. And we were really interested in looking at how trabecular bone in these early fossils compares to ours and compares to Australopithecines.
What goes into the process of analyzing a fossil?
So there are multiple things. First, getting access to the fossils is a first step. And then when you get access to getting permissions from different museums or curators that hold these collections, is to get access to a high resolution micro CT scanner. And often these things are usually in different parts of the world. So the collection can be in country A and the scanner might not be in that country, and you have to take the fossils elsewhere. So part of the project that I was mentioning that the Leakey Foundation funded is the study of fossils from Kenya of early Homo and Australopithecines that we are going to carry, or we're going to transport to South Africa for scanning, and then spend days and days, many hours scanning these fossils because it's very time intensive.
It's so cool. It's so neat to
from micro CT imaging. Then
oh, sorry, I think there was.
Yeah, it is, and one of those pictures you just showed. I'm sorry.
Oh, sorry. No, go ahead. I think there was a bit of a delay there, but yeah, please go ahead.
Yes, and one of those pictures that you just showed is of the micro CT scanner. It's a huge piece of equipment that we prepare the specimen and put it in the scanner, and you leave it for sometimes even 24 hours for it to scan in 3D dimension and assemble all the individual X-rays to produce a lovely 3D rendering. And yes, and I'm glad you brought this picture up because it shows us an example, after we've got the images. So this is an example of a bone that we are analyzing in the lab. So that's a hand bone that's sitting there on the screen. And we will use the different programs we use to process, to clean, and then look at the inside of bone and then quantify the amount of bone that's in there.
What are some of the biggest factors that make analysis difficult, and how do you account for that when analyzing?
Yes, so that's a really good question. So when we are dealing with scanning of fossils in particular, sometimes the bones may be highly fossilized, that it's hard to visualize what's inside, what's bone and what's non-bone. And that's a beautiful picture of one of the fossils from Kenya, of early Homo, that shows you the little stripes that you see in gray, that's bone. The lighter shade is non-bone. So it's just all the soil, the minerals that have been sitting there for millions of years. So that's usually our challenge. The biggest challenge is to be able to clean out the non-bone material so that you can only have bone and quantify that. So some people that are very clever, Chris Dunmore and colleagues, have come up with ways of analyzing this that have been helpful. But sometimes if it's really dense, you can't make out what bone is versus non-bone, then there's not much you can do. So that's really one of the biggest challenges. The other thing I guess I can mention is that the cost of scanning is quite high. So things have improved over the years, and more institutions have scanners now. But these can, drive the cost. The costs are quite high that you might not be able to scan everything that you need. It's also time consuming, so that's another challenge. Well, and more recently, of course, the pandemic has stopped, has put travel to a halt. That it's getting harder and harder to go from country to country and scan these fossils.
Well, yeah, and you have to, balance, but bringing the scanning to the fossils or bringing the fossils to the scanning.
Yes, exactly.
So I'm really excited to hear more about your research. If you want to start sharing your slides before we turn the virtual floor over to Habiba. If you are enjoying this episode of Lunch Break Science, be sure to subscribe to our YouTube channel or follow us on Facebook and Twitter to be the first to hear about upcoming episodes. So now let's turn the virtual floor over to Habiba.
Do you see my screen, Ariel?
Yes, we see it.
All right, excellent. And, just move this a little bit. All right, so thank you very much for the opportunity to share some of my work with you and with the audience. And I'm basically going to give a brief overview of what my research interests are and a few results that we have found so far. So as I probably alluded to during the interview session of this of our conversation, I'm interested in how activity, and in this case we can refer to it as behavior, influences bone morphology. How does what we do affect our bones? And I have put up a few 3 pictures here just to show you what I'm working with. So I'm working at we are looking at these the picture on the left here is trabecular bone in the femoral head or in the thigh bone of a fossil ancestor. And then we have one of the fossil ancestors, in particular, this is Turkana boy. And then I'll give you a glimpse of some of the work that comparative work that we are doing that has to do with it has to do with making comparisons across carnivores, canids in particular, the dogs. So to start out, we know that bones can tell us a lot. They can tell us about someone's age or population's age, whether it's humans, whether it's other animals. We can tell sex from the skeleton. We can estimate stature, how long a person was. We can infer pathologies. Did an individual have, arthritic bones? Did they have perhaps osteoporosis and things like that? Another pathological cases, or if a population had pathologies. We can infer diets from bones, and we can infer how much activity a population was engaged in. And of course, we can tell or infer evolutionary change through the study of bones. And what I'm particularly interested in is how physical activity or mechanical loading, how much stresses we put on our bones affects our bones. And then I'm also particularly interested in how we can tell evolutionary changes over time by looking at the human fossil record. So some of the things that we know already from research from over the last 100 years and slightly more is that bone generally increases in density in response to strain and stress. And when I say increasing in density, I mean there will be more bone growth in a part or a region of our skeleton that we are using the most. There will also be changes in the alignment of trabecular bone in particular. And this means that trabecular bone is, all bone is informative, but trabecular bone is quite informative because it not only increases in density, but it also aligns itself in the direction of strain. So let's take a look at these pictures. So these are sections, microscopic section. These are sections through the thigh bones of a fetus, then infants, up to a toddler. And what you're looking at the fetus here, if you're looking at their femoral neck in particular, where at the hip joint, you see that there's a blob of bone. The white material is bone. By about 6 to 12 months, you see that there's more spacing in there. There's more black, which is the space, but there's the bone itself is a little bit more spread out. By the time a child is a toddler, we see an interesting pattern that's very striking, and that is the bone, trabecular bone, is aligning itself in this manner from up, down, up, down. And this is interesting because it coincides with the time that children are beginning to walk, meaning as they walk upright, they're putting strain on that femoral neck. And consequently, the bone is also responding by growing or by aligning itself to resist those compressive and compressive forces and tension. So just to back up and to summarize a little bit, I'm interested in changes that occur within a lifetime, such as the ones we just saw in the previous slide, but I'm also interested in changes over a long time. So this chart here shows us the current state of affairs or state of fossils, I should say, when it comes to the human fossil record. Although, of course, more recently, a few others have been added up here next to Homo sapiens and Neanderthalensis, if you've been following the news. So, but right now we have this as it is. But what I'm interested in when it comes to the skeleton, the gracility or changes in bone, I'm interested in what's been happening from about 2 million years, maybe 2.5 million years to the present. And that's an interesting time because that's when we see the emergence of the genus Homo. That's our genus. And that's when you also see a great proliferation of different species of Homo, as well as Australopithecines. So going back or referencing that slide about the emergence of our genus in the last 2.5 million years, we know that there are some features that emerge that far back in time and that are persistent to the present. And some of them, we are referring to them as uniquely modern human features. And those include traits that are related to our ability to walk upright, such as having a narrow pelvis, such as having large joint surfaces that can support our body weight, such as having short toes, and possibly even long legs, although there's been a debate about if this is truly a modern human feature, so to speak. And more recently, it seems that we also have we, in this case, being modern humans, that we have a uniqueness in having gracile skeletons or lightly built skeletons, in having low bone density. So the question is, are the gracile skeletons we are seeing a result of changes in activity levels, or is it an evolutionary adaptation? And while that question might seem simple and straightforward, it's actually not, because to test this type of question will require a lot, a lot of work to be done in the fossil record, comparative work, a lot of comparative work in modern, not just modern humans, but in modern mammals that have traits similar to ours. And so this is, of course, work that is continuing, that I'm still working on and will likely be working on for a very long time. So I've touched on the question, but let me get to the more specific question that I'm interested in, and that is how to get to that, or to get that bigger question of, is this a change within because of a low activity or because of due to evolutionary change? We first have to sample our evolutionary timeline and look at all the fossils that we can find and see if the pattern of low trabecular bone density or the gracile skeletons that I'm mentioning, if these are indeed a recent evolutionary event. Did they happen in the last 10,000 years? Did they happen in the last 100,000 years, in the last million years, or as far back? Or how do they compare? How do the recent human ancestors compare to the older human ancestors from as far back as the Pliocene and the Pleistocene? And I give the dates there for reference. Now, that's one question. The other question is, if this gracileization is happening only in the last 10,000 years, as my research seems to suggest, although not totally conclusive, it's not complete yet. If that's the case, then what's causing this gracilization? Is it because of the so-called human self-domestication hypothesis? And I'll touch on the human self-domestication hypothesis here in a little bit in the three slides down the line. And that's the idea that there's evolutionary selection for group cooperation in humans whose byproduct may be gracilization. So to answer that question, of course, as we had talked about earlier on, I'm interested in looking at bone density, and I've thrown some of these pictures up that you are shown, and that's showing I'm preparing a bone, a femur. This is a femur bone at the hip joint. I'm preparing it, getting it ready for scanning in this micro CT scanner. So there I am holding the little bone, and this is an early Homo bone from Kenya, from northern Kenya. We put it inside a piece of foam to stabilize it and have it in a position that when X-rays are run through it, we will get a clear picture or a good image from it. And here is an example of a micro CT scanner. This one is a micro CT scanner from the American Museum of Natural History. And then in the bottom slides, I have some examples of specimens that we have already scanned, or we got access to them through the Max Planck Institute. So, for example, skeletons from about 26,000 years ago from the Czech Republic, the Dolní Vestorniči skeletons. Some others also from about the same time period from Israel, Ohalo. And then you have this early one from about 2 million years from East Africa. And then the last image here is just to show you how we quantify trabecular bone. So when I'm talking about trabecular bone density, you might be wondering, oh, how do you arrive at that? So after scanning our bone here, we end up with an image that we can reconstruct to appear like this. And then we have a program, we read the image into a program. There are several programs that we can use, and we can visualize the inside of bone as a section. And what we do is take a region of interest that I show in pink here, and we quantify the amount of white pixels. In this case, white pixels represent a bone, and we divide that to the total volume, so the total number of pixels. And we end up with a value, a ratio that we refer to as BVTV. And that's what I'm referring to by trabecular bone density. So trabecular bone density doesn't mean the mineralization of bone, but the amount of bone in a joint. Now, with that in mind, let me just give you a quick overview of some of the specimens we have scanned, some of them and others that we are yet to scan. So the ones in blue are those that we have already scanned. Some of them we've published already. Some of them we are still working on. So we have Australopithecines from South Africa, early Homo from East Africa, early modern humans from Europe and the Levant and the Middle East, Neanderthals, upper Paleolithic humans from Europe. And then we have modern human collections of Native Americans that were foraging, that were agricultural. We have very recent modern humans from the US. We also have an Egyptian collection that was agricultural. So we are yet to study the ones in black. So there's some Paranthropus, Neanderthals from the Middle East, as well as hunter-gatherers from South Africa, from Australia, and from South America, Tierra del Fuego. So all these will form that, it's the attempt to try and sample as many hominins or as many human ancestors as we can, at the same time comparing them with modern humans. So I'll share with you here one of the results, or one of the box plots that we have prepared, comparing humans, or early Homo to be precise, and Australopithecines, as well as modern humans. So what we are looking at here is one specimen of early Homo, because the work is not yet complete. That's from East Africa. And then we have Neanderthals, early modern humans from the Middle East and Europe. Then we have those early Paleolithic humans I was talking about from the Czech Republic. And then we have modern humans, that is the Native American ones I was talking about, and the Egyptian ones I was talking about. And what's striking is that this group of recent modern humans have significantly low trabecular bone density compared to all the other members of early Homo. So even of not just early Homo, but Neanderthals, early modern humans, and upper Paleolithic humans. Then if we include the Australopithecines, so Australopithecus sediba, Africanus, and this one that's not assigned to any particular species, but it's considered an Australopith, you see that they all also have high bone density, close to those of the other Homo species. So the striking difference is in the recent modern humans. So the conclusion here seems to be that these gracile skeletons, or the low bone density in modern humans, is a result of reduced physical activity. Because if I may go back, all these are early in time. These are the ones from the last 10,000 years. So in the last 10,000 years, we know that that's the time when we started domesticating crops, we started domesticating animals, started living in communities, and later on, of course, in city-states and towns and so on. Well, these other guys are still either hunting, gathering, climbing, if you will, just like our closest living relatives. So the other thing that I would like to bring to your attention in terms of that gracility I'm talking about, not just in the graphs, but if we look at the insides of bone, particularly in the hand bones, what you will notice is that in recent humans, we have very sparse bone. In early modern humans, from about 100,000 years ago, we have sparse bone, but not quite as sparse as ours. In Australopiths, you have dense bone, and in chimpanzees, you have dense bone. So it seems to be something that's happening late in time. But again, I say this with a bit of caution because we are yet to sample the whole of the Pleistocene and the Pliocene. So still more work to do, but it seems this is the pattern for now. So if that's the pattern, that we are seeing a change in the last 10,000 years, why is that the case? Where is this gracileization arising from? Is it just what I said? It's because of reduced activity, because we are living in sedentary societies? Or is there something else that's taking place? So that's why I asked the question, is the observed low trabecular bone density a result of self-domestication? And this human self-domestication hypothesis posits or postulates that as we became we became more cooperative in the last 10,000 years, and part of that cooperation and friendliness, or what we refer to as prosociality, resulted in gracile skeletons. So it wasn't a selection for gracile skeletons, but a selection for friendliness that without, which led to these gracile skeletons. And this makes sense because if we look at animals that have self-domesticated, and in particular, if we look at the dogs, domestic dogs compared to the wolves, we see gracileization of the crania. So if we look at the wolf here, and then we have the dog here, we see that generally the crania have become more gracile, so there's not much rugosity. We see that the cranial capacities have reduced in domestic dogs. There are changes of differences in snout length, and so on and so forth. And these are things that have been studied quite extensively. So if we think about that, and then pull in this evidence that came from the silver fox experiment, which probably many of you are familiar with, and I'll just quickly summarize this experiment. This was an experiment that was done since the 50s in Siberia in Russia, and basically silver foxes were bred, tame silver foxes was bred in one line lineage, and wild ones, or aggressive ones to be more precise, were bred in another line. And this behavior, being aggressive or tame, was the one that's being selected for, artificially selected for. But it turned out that it resulted in changes in morphology, so such that the aggressive strains of these foxes tended to have larger teeth, they tended to be a bit more robust in cranial features compared to the tame line, which tended to have smaller canines. And there are other morphological features that are such as changes in coat pigmentation, that the tame line ended up with the pigmentation of the coats. They ended up with floppy ears, curly tails. In other words, they became more like dogs compared to the aggressive strain, which reinforced the idea that certain behaviors seem to result, or selection for certain behaviors results in changes in morphology. So could something like this be happening in humans? Could this self-domestication of humans, being more cooperative, working together with one another instead of aggression, be resulting in gracileization? But of course, before we go to that big question, we first need to establish a pattern in dogs. And so what we've been doing is comparing domestic dogs to wolves, and in particular looking at trabecular bone density to see how gracile they are. So we've made comparisons of the great wolf and several breeds of dogs. And as you can imagine, of course, dogs will vary quite extensively depending on what they've been bred for. So we have Eskimo dogs, huskies, St. Bernards, greyhounds, and dingoes, actually, from the archaeological record. So I'm going to show you the preliminary results that we published recently comparing gracilization of wolves and dogs. And here we are looking at the bone density in the femoral head. And this is the wolf, and this is the dog. And just to remind you quickly that the bold line of these box plots refers to the median. The tails here, the whiskers refer to the range. So overall, even if the domestic dog has a wide range, as you can see, it is more gracile, actually significantly more gracile compared to the wolf. So meeting or supporting the hypothesis that they are more gracile. So the other thing that probably you might have a question about is that it's there's a wide range here. And yes, there is a wide range because, of course, we've pulled together all the different breeds of dogs. And you have a greyhound, which is quite gracile, and you have an Eskimo dog, which is quite robust generally. So that creates that variation. So to summarize, basically what I've shown you is that humans have gracile skeletons, and this seems to be a recent phenomenon. And this is work that we've been as a reason from the comparisons of early Homo, early modern humans, Neanderthals, and other upper Paleolithic Homo. Secondly, that domestic dogs are more gracile compared to their wild counterparts, the wolves, which is potentially a result of the domestication process, and which probably might end up explaining that gracilization beyond just activity. And the other thing, of course, to keep in mind in these types of research is that there's the bigger goal of trying to understand the implications of evolutionary adaptations on bone disease, right? If we've evolved, or we have these gracile skeletons that are occurring late in time, is that the beginning, or is this a over-selection, if I may use the term, of gracile bone that's resulting in present-day issues such as osteoporosis? So my future work and work that I'm continuing to do is plentiful. We still need to document a lot of changes that are related to the domestication process. So with that, I would like to thank all of you for being out there listening and watching. I would like to thank Ariel for inviting me, and of course the Leakey Foundation for giving me this opportunity to speak, and as well thank all the funding institutions, including again the Leakey Foundation, my own institution, Marshall, the NASA itself, the American Association for Anatomy that have all funded my work. I would also like to thank institutions that have allowed me to access to specimens, or allowed me to scan using their facilities. NEXA in South Africa, access to specimens through the Smithsonian, access to specimens through the Max Planck Institute, curators, David Hunt, Darren Lundy, Eleanor Hoger, Job Kibi, and Carol Ward. All of these people that have been fantastic around my work. And of course, last but not least, my students that have been, that continue to do research in my lab and allow me to, be able to give a talk such as this. So thank you very much. I hope I haven't gone over time too much.
No, that was perfect. Thank you so much. Do you want to tell us a little bit about your upcoming projects? Before we're going to get to questions from the audience pretty soon, but I figured that would be
yes.
I want to hear more.
Yeah. So there are two things, and I don't know if I have anything to show you here. Yes. I have one thing here. So there are two things going on: the fossil, human fossils project, and then the canid project. So here's one example of work that we haven't finished the analysis, comparing different, not just wolves and dogs as I showed, but including the dingo, including other canids. Think of the African hunting dog, think of the jackals, think of the foxes. We haven't done that yet, coyotes. And this is just a broad comparative set of data looking at the differences in gracility or robusticity, if you will, across canids. Then the other work is the work that I mentioned, which is supported by the Leakey Foundation, that is looking at early Homo specimens and fossil specimens and Australopithecines, and we would like to scan as all these long bones that we have that are available at the museum in Nairobi, and look at the patterns of gracility or robusticity for that matter. And again, the pandemic has made that very difficult to do. So we'll see how next year goes. Maybe we'll have more data. So that's a few of the ongoing projects that I have.
And beyond the scope of your research, you also do science outreach. Would you tell us a little bit about your outreach?
Yes. I did not include that, but we have been doing some outreach, teacher outreach in Kenya in particular, along with Brianna Pobina at the Smithsonian. And basically, we are using materials that have been developed by the Smithsonian to give teachers access in Kenya for them to teach evolution in their classes. So they'll teach evolution, right, as the textbook says, define evolution and what have you. But we are preparing these materials, mostly pictures, because getting access to casts of fossils is quite pricey. So mostly pictures that we've laminated, and we are taking them out to the teachers, putting them in binders and folders and so on, and then introducing the concepts of ways of teaching evolution in a way that can be more engaging with students. So it's not something you just hear about, but you can relate to. So that's one way of doing that. Then the other one has been what we refer to as teaching evolution with religious sensitivity, or with cultural sensitivity, so that you're teaching evolution not from a point of, well, evolution and science are not do not go hand in hand or cannot sit together, some people say. Not that I'm objecting to that, but in a way that people can appreciate evolutionary change within their religious context, if they like. Right. And that's also work that's been done a lot by the Smithsonian. So the last 2 years, well, not last year, but 2018 and 2019, we were able to travel to Kenya and we did 2 workshops with the Kenyan teachers, high school biology teachers.
If you're watching and are interested in hearing more about sharing human evolution with the public, we had a lecture last week with Habiba's colleague at the Smithsonian, Brianna Pobiner, and we've shared the link to that talk in the chat. So definitely check that out. Now let's take some questions from the audience. We already have a couple of them, so that's always kind of exciting. Our first question, let's see here. H asks, could you tell us a little bit about how the COVID-19 pandemic has affected your research activities and those of your colleagues?
Oh, yes. I start with because it really has. For instance, I'll just tell you the one, because we are in a Leakey Foundation forum, I'll tell you the work that was funded by the Leakey Foundation. We were meant to start it last year. And of course, the pandemic hit, we could not travel. And we were meant to travel to Kenya, get our fossils from there, along with my collaborator from the museum in Nairobi, and take those fossils down to South Africa, scan them, then return them to Kenya, and then come back to the US with data to analyze. But we haven't been able to travel to any of these countries because, of course, at the point where we were ready to go, then there were new variants of the virus coming up in South Africa. The infection rates were quite high, so we are sitting still. So really, this has put us quite behind in terms of research, because at the earliest, probably we'll be able to do this next year, hopefully. Vaccines being available, hopefully in the rest of the world also being an issue, being a thing that we hope will be achieved soon. So yes, it really has been challenging. But on the other bright, it's not all doom and gloom. On the other side, on the other hand, we still have other things, local projects going on that relate to existing data. For example, those canids I was talking about, scan. So analyzing them in the lab. So and I think that's the same thing, since you asked about colleagues also, it's the same thing across the same problem other paleoanthropologists are experiencing in terms of doing field work. I know folks that usually go to East Africa for field research or South Africa for field research, and they've just not been able to.
Our next question, let's see here, it comes from Anne-Marie. She asks, could upright walking have contributed to gracileization in humans? Can a direct relationship be established?
Well, yes and no. Well, I would say it's a good question. It's a challenging one. So when you think about gracileization and upright walking, right, you think about the upper limbs being more gracile in humans, because we are not using them anymore for climbing and for arboreal locomotion, but the lower limb, we're using it in bipedal behavior. It's being used extensively, so there'll be robusticity there. So in a way, depending on which limb elements you're looking at, yes, you can see differences in gracileization and robusticity. But then again, when we look at Australopithecines and you compare them to early Homo, so if you're thinking Australopithecines probably were doing both bipedal and arboreal behavior, you see they're actually quite robust in both upper limbs and lower limbs. Then we look at Homo, they are robust in the lower limb. So in that way, you can make that link. But you throw in some species such as Australopithecus sediba, then you get an interesting pattern. You see a mix of features of robusticity in the tibia. One of my data points on the slide showed that. But in the hand, you see both tibia and in the hand, you see robusticity. But when it comes to the femur, you see a bit of gracileization. So yes and no, I suppose. Not, there isn't quite a straight line. And that's why the more sampling we can do, probably we can answer some of these questions a little bit better.
Our next question comes from, let's see here. Oh, Walter, have you seen osteoporosis in any of your study specimens? And there's actually a second part to the question. Have you seen bone pathology in your study specimens, like intrinsic bone tumors, Paget's disease, and cartilage disorders?
Yeah, so when we look at, I'll start with the fossils. In the fossils, it's hard to recognize, especially osteoporosis, right? Because these bones are highly fossilized. So it's a little bit of a challenge. But there are people that have identified, colleagues that have identified pathologies in some of the fossils. I haven't seen it, but mostly what I've seen is, I think, not tumors, but bone growths around the outside of bone. So a little bit. When it comes, so in reference to trabecular bone for the fossils, no, I haven't seen any, but it doesn't mean it's not there. It also could be a result of just the fossilization, the extent of fossilization. And when it comes to modern humans, the comparative samples, yes, you do see after the age of 45, 50 and above, if the known ages are there, you'll see some bones are quite porous. So whether that's, yeah, that could be osteoporosis in some of the very modern collections, for example, the ones from 100 years ago. You can tell that. But for those that are, for those that are from the archaeological record, there's also leaching of bone for having been in the ground for a long time. So they might be light, not necessarily because of bone disease, but because of leaching. Then there were a couple other diseases he mentioned. The person asking the question mentioned, but for in the ends of in the joints on the outside of bone, you can actually see, yes, sometimes you can see arthritic bones or bone growths around the joints. And that's in modern humans, but no, I haven't seen any in the fossils.
I have a question kind of to add, thinking about breaks and fractures with gracileization. Do you see more breaks and fractures, or how is that kind of reflected?
Yeah, so that's those two with the previous question, they go hand in hand. So it's hard to say from the fossil record, again, because preservation is a big issue, and bones that are a little bit more dense tend to be preserved. The ones that are lighter will be destroyed, or will not find them. So it's hard to say in the fossil record in terms of cracks and fractures. But in the archaeological record, yes, you can see, again, fragile bone or bone that seems overly gracile. But just to go back to in terms of how we sample, when you see something is too porous, then you don't include it in the study, just to make sure you're avoiding any biases that may occur, whether it's due to leaching or actually due to bone disease. So we usually avoid those. And if it's known age, of course, we try to cut it off at 45 years of age and below.
Our next question comes from John. Why compare humans to canids as example of domestication as opposed to other domesticated group like felids, bovids, and the like?
Great question. It's not excluding the others. It's just the start of it. And dogs seemed like the ideal self-domesticated species. It's domestic dogs. And the logical thing was, well, let's look at domestic dogs compared to wild, their wild counterparts, all kinds of species. The next thing, actually, which I did not mention at all, is that we are interested in cats too, and we have data on different wild cats, panthera, leopards, jaguars, mountain lions, that we would like to also pursue the same questions with domestic cats. So that's the next step. He also mentioned bovids. Yes. So there are two angles. I should back up a little bit here. There's two ways of looking at these. There's the self-domestication, the animals that underwent self-domestication, and then there are animals that were that humans domesticated. the like active domestication, right? So in the case of bovids, for example, think about cows compared to the wild, the domestic cow and the wild cow. So though that's also another angle to look at. The challenge still is, of course, how to reconcile the two, because if we are thinking about domestication that's driven by humans, that's actively, there's active selection by humans. Humans are selecting for larger animals, right? A larger goat, a larger cow, and so that might have an influence on robusticity. That's not the self-domestication we were talking about. So, but these are certainly great ideas, avenues to pursue.
My last question for you is, do you have any advice for those who are watching who are interested in studying paleoanthropology as a career?
Right. Well, I would say, like, I suppose, as it was said to me before, is that you follow your, I know it sounds very cliche, it's follow your heart, follow your passion, if you will. Think about, but also very important is to think about the careers that you can get into. And some careers in paleoanthropology will require PhDs, some will not require PhDs. So follow your passion, think about the future in terms of employment or earning, and then make your decision based on that. And that way you can also decide, well, if it's a PhD, you want to look at which PhD programs specialize in paleoanthro, what kind of paleoanthro do I want to study, and so on.
Well, thank you so much for joining us, Habiba. Next time on Lunch Break Science, we meet primatologist Melissa Emery Thompson and learn about the life histories of the Kanyawara chimpanzees of Kimbale National Park in Uganda. The next program in our summer travel series is on July 7th at 10 a.m. Pacific. You'll be virtually transported to Saudi Arabia for a behind-the-scenes tour of the earliest known Arabian archaeological sites with Leakey Foundation grantee Dr. Michael Petraglia. Master falconer Kate Martin also provides a glimpse into the ancient Saudi Arabian hunting tradition of falconry. Attend this program live for a $100 donation or receive a link to the program recording for a $10 donation. All donations to the Primate Research Fund will be quadruple matched, thanks to generous sponsors. You'll protect vulnerable primates, long-term field research sites, and their local staff members, have an exclusive behind-the-scenes look at some pretty amazing locations, and much more. We've shared the link to this program in the chat. So I hope you'll be able to join us for these amazing events coming up. And again, Habiba, just thank you so much. We really just enjoyed having you on.
Thank you so much for inviting me and having me. And thank you, everybody, for taking the time to listen to this.
Well, thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next time, stay hungry for knowledge.
Bye-bye.
Lunch Break Science is brought to you by the Leakey Foundation and made possible by the generous support of the Anna Gordon Getty Foundation and Camilla and George Smith. Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, today's guest scientists, and how you can help support human evolution research and educational programs like Lunch Break Science. Right now, all donations to the Primate Research Fund will be quadruple matched by generous donors, meaning your impact will be quadrupled. Miss an episode of Lunch Break Science? Catch up on past episodes and browse our library of Leakey Foundation lectures on our YouTube channel. Still hungry for science and can't wait till our next episode? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Subscribe to Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about exciting upcoming episodes and programs, as well as groundbreaking discoveries in human evolution research.